Introduction to the Cell. Welcome to this quiet space of learning. Tonight, as you begin to settle in, as you let the day's concerns drift away, we will begin our journey into the world of biology. This is the study of life itself, in all its complexity and all its simplicity. And at the very heart of this study, at the very foundation of what it means to be alive, is a single, profound concept: the cell. Our first lesson tonight is an introduction to this fundamental unit of life. There is no better place to begin our understanding of the vast, intricate tapestry of the living world. So, as you find comfort, as you allow your breathing to slow, let your mind be open to this first, essential lesson. What is a cell? We can use simple analogies. We might call it a tiny factory or a microscopic city. And these are helpful, but its true definition is even more fundamental. A cell is the smallest structural and functional unit of an organism. It is the minimum amount of stuff that can be considered, in and of itself, alive. It is a self-contained, self-sufficient, and self-replicating entity. It is the building block of all life. Just as an atom is the basic unit of matter, the cell is the basic unit of life. Let that idea settle in for a moment. Every living thing you have ever seen, a tree, a bird, an insect, or your own reflection in a mirror, is composed of cells. Some organisms, like bacteria, are just a single cell. Others, like a human, are a vast, organized community of trillions of cells working in harmony. All cells, regardless of where they are found, share a few key features. They are all enclosed by a boundary, a skin called the plasma membrane, which separates the inside of the cell from the outside world. Inside this membrane is a jelly-like substance called cytoplasm, where all the internal machinery of the cell is suspended. And all cells contain DNA, deoxyribonucleic acid, which is the genetic blueprint, the instruction manual for everything the cell is and does. Though all cells share these basic parts, they are not all the same. In the vast history of life on Earth, two major designs have emerged. We call these prokaryotic and eukaryotic. Let us first consider the simpler of the two, the prokaryotic cell. The name itself offers a clue. Pro means before, and karyon means kernel or nucleus. These are the before nucleus cells. They are ancient, the first forms of life to appear on our planet billions of years ago. Organisms like bacteria are prokaryotes. Their structure is simple. They have no nucleus. Their single circular strand of DNA simply floats within the cytoplasm in a region we call the nucleoid. They also lack the complex, membrane-bound internal compartments that we will explore later. They are simple, but incredibly successful and resilient. If you are still awake, picture this simple prokaryotic cell. A membrane, some cytoplasm, a floating strand of DNA, and a few other small parts. It is a model of efficiency. Now, let us consider the other design. The eukaryotic cell. AU means true. These are the cells with a true nucleus. Eukaryotic cells are much larger and far more complex. They are the building blocks of all the life you can see with the naked eye. All plants, all animals, all fungi, and the tiny protists. The defining feature of a eukaryotic cell is its nucleus. This is a membrane-bound command center, a secure vault that houses the cell's DNA, protecting it and organizing it into structures called chromosomes. But the complexity does not end there. Eukaryotic cells also contain an entire suite of smaller, specialized compartments called organelles. Each organelle performs a specific job, allowing the cell to function like a miniature city with different departments. There are power plants, factories, shipping centers, and waste disposal systems, all working together. This distinction between the simple prokaryote and the complex eukaryote is the most fundamental division in all of biology. The knowledge of the cell is not something we have always had. It was a profound discovery, built over centuries by patient observers. In the 17th century, A scientist named Robert Hooke used a primitive microscope to look at a thin slice of cork. He saw a honeycomb of tiny, empty boxes, which he named cells because they reminded him of the small rooms in a monastery. He was seeing only the dead, empty walls of plant cells. Around the same time, Antony van Leeuwenhoek, a Dutch cloth merchant, crafted powerful single-lens microscopes. He turned his lenses to pond water, to dental scrapings, and was astonished to find a world of tiny, living creatures, which he called animalcules. He was the first human to see living, single-celled organisms. It took another 150 years for the full picture to emerge. In the 1830 seconds, the botanist Matthias Schleiden declared that all plants were made of cells. Shortly after, the zoologist Theodor Schwann declared that all animals were also made of cells. These observations formed the basis of the cell theory. This theory, as it was further refined by the scientist Rudolf Virchow, rests on a few foundational pillars. First, all living organisms are composed of one or more cells. Second, the cell is the most basic unit of structure, function, and organization in all living things. And third, in a phrase that ended the debate on spontaneous generation, omnis cellula e cellula, all cells arise from pre-existing living cells. Life begets life, and it does so at the cellular level. Why is the cell considered the smallest unit of life? This is a crucial point. A cell can do everything required to be alive. It can perform metabolism, turning nutrients into energy. It can grow and respond to its environment. It can maintain a stable internal balance, a state called homeostasis. And, most importantly, it can reproduce, passing its genetic information to the next generation. An organelle, like the nucleus, cannot do this on its own. A virus, which is even smaller, cannot. A virus is just a package of genetic material that must hijack a living cell to reproduce. Only the cell, as a complete system, has all the parts necessary for independent life. As you rest, consider this. Your body, the one you are resting in right now, is a testament to this theory. You are a living community of an estimated 30 trillion eukaryotic cells, and you began as just one, a single, fertilized cell that divided and divided and divided again. As these cells multiplied, they differentiated, taking on specialized jobs. Some became nerve cells to carry thoughts, some became muscle cells to create movement, some became skin cells to form a protective barrier. Yet all of them, from the lens of your eye to the bone in your toe, are cells. They are all descendants of that first cell, and they all work together to create the complex organism that is you. Understanding the cell is the foundation of medicine, of genetics, of all life science. We have now laid the groundwork. We have defined the cell. We have learned of the two great types, prokaryotic and eukaryotic, and we have understood the cell theory. To truly understand how a complex eukaryotic cell functions, we must now move past the outer wall. We must go inside. We must look at its internal components, the specialized organs of the cell. These are known as organelles. In our next lesson, we will begin this internal exploration. We will start with the most important organelle, the command center that defines the eukaryotic cell, the nucleus. But for now, just rest. Let these foundational concepts of life settle into your mind. The cell, the unit of life, the beginning of our journey. The nucleus and genetic control. We now continue our quiet journey, moving from the general concept of the cell to its inner world. In our first lesson, we established the cell as the fundamental unit of life, and we distinguished between the simple prokaryote and the complex eukaryote. Our focus now is on the complex eukaryotic cell, the type of cell that makes up your body, and all plants and animals. Our next module focuses on the single most prominent, most defining feature of the eukaryotic cell. We are moving inside, past the cell membrane, to explore the administrative center of the cell, the nucleus. As you settle deeper into a state of rest, you can picture the cell as a vast and busy city. If the cell is a city, the nucleus is its central government, its city hall, and its grand library, all in one. It is a large, typically spherical organelle, and it holds the most precious possession of the cell, the genetic blueprint for the entire organism. Let us first examine its structure. The nucleus is not simply floating unprotected in the cytoplasm. It is enclosed by a remarkable structure called the nuclear envelope. This is not a single wall, but a double membrane, one layer inside the other. This double layer acts as a highly secure barrier, protecting the priceless DNA inside from the chemical chaos of the rest of the cell. This envelope, this boundary, is not impermeable. That would be useless. It is studded with thousands of tiny, intricate gateways called nuclear pores. These pores are not simple holes. They are complex molecular machines, security checkpoints that actively and carefully regulate what passes in and out of the nucleus. Raw materials, like the proteins needed to build ribosomes, are actively imported into the nucleus, and the instructions we will soon discuss are actively exported out. Inside the nuclear envelope, we find a jelly-like substance called the nucleoplasm, which is similar to the cell's cytoplasm, but specific to the nucleus. And within this nucleoplasm, we can usually see a dark, dense, non-membrane-bound region. This is the nucleolus, which means little nucleus. You can think of the nucleolus as a specialized factory. Its one and only job is to manufacture the components of ribosomes, the cell's protein-building machines. The nucleolus is the factory that builds the other factory workers. We will discuss those ribosomes in a later lesson. But the most important content of the nucleus, the very reason for its existence, is the cell's genetic material. This material is DNA, or deoxyribonucleic acid. The DNA molecule is a long, twisted double helix, a ladder of information. It is the master blueprint, the complete instruction manual for building and operating the cell. The sheer amount of DNA in a single human cell is staggering. If you were to unwind all the DNA from one nucleus, it would stretch nearly 2 meters, or about 6 feet. How does the cell fit 6 feet of DNA into a microscopic nucleus? It does so through brilliant organization. The DNA is not just dumped inside. It is tightly wrapped, coiled, and spooled around proteins called histones. This combined structure of DNA and its organizing proteins is called chromatin. For most of the cell's life, the chromatin exists as a long, tangled, thread-like mass, like a giant, chaotic ball of yarn. This messy state is actually functional, as it allows the cell's machinery to access specific parts of the DNA to read the instructions. However, when the cell prepares to divide, it must make a perfect copy of its DNA and then distribute it evenly to 2 new daughter cells. To do this, the tangled chromatin undergoes a remarkable transformation. It condenses, it coils upon itself, folding and packing into the dense, rod-like structures that we can see with a microscope. These dense, packed-up structures are what we call chromosomes. Think of it this way. Chromatin is like a full set of blueprints spread all over a giant room. A chromosome is those same blueprints, all neatly copied, collated, and bound into a set of books, ready for transport. Let this concept settle. In the nucleus as a library, with chromatin as the readable, in-use books, and chromosomes as the same books, packed tightly in boxes, ready to be moved. We call the nucleus the command center, or the brain of the cell. But how does it command? It doesn't think. It controls the cell by controlling its proteins. The DNA in the nucleus contains thousands of recipes called genes. Each gene is a specific segment of DNA that holds the instructions to build one specific protein. The nucleus protects the master blueprint of the DNA at all costs. It never, ever lets the original DNA leave its secure, protected walls. So, when the cell needs to make a specific protein, say, an enzyme to digest food, it must consult the blueprint. The process is one of elegant delegation. Inside the nucleus, a molecular machine finds the correct gene on the DNA strand and creates a photocopy of it. This photocopy is not made of DNA. It's made of a similar single-stranded molecule called messenger RNA, or mRNA. This process is called transcription. This mRNA message is a temporary, disposable copy of a single recipe. It is small and mobile. It is allowed to exit the nucleus, passing through one of the nuclear pores. It travels out into the cytoplasm, carrying its instructions. Once in the cytoplasm, the ribosomes, the very machines built by the nucleolus, will read this mRNA message and begin to build the protein, amino acid by amino acid, exactly as the gene prescribed. This is the central function of the nucleus. By controlling which genes are transcribed into mRNA, the nucleus controls exactly which proteins the cell makes, and when it makes them. And because proteins are the workers of the cell, forming its structure, running its chemical reactions, and acting as signals controlling proteins, means controlling everything. Cell growth, cell metabolism, cell specialization, and, ultimately, cell reproduction, all are governed by the nucleus. This genetic information stored in the nucleus is the basis of heredity. The DNA in your nuclei is the instruction set that makes you, you. It determines your inherited traits, from your eye color to your blood type. It is a complete manual for building a human, passed down and recombined through generations. This is the entire foundation of the field of genetics. We have now explored the nucleus, the command center. We understand its structure, the envelope, the pores, the nucleolus, and its precious cargo, the chromatin, which condenses into chromosomes. We understand its critical function, to protect the DNA and to control the cell's life by sending out instructions in the form of messenger RNA. The nucleus holds the instructions, it writes the work orders. But all of this building, all of this copying, all of this controlling, all of this requires a vast amount of energy. The nucleus holds the plans, but it does not create the power. To carry out these complex instructions, the cell needs a reliable, constant, and powerful energy source. Our journey must therefore lead us to the power plants of the cell. In our next lesson, we will explore the organelles responsible for creating the energy that powers all of life's work. We will explore the mitochondria, but for now, just rest. Let your mind absorb this knowledge of the cell's elegant control system, the nucleus, the library of life, mitochondria, and energy production. In our last lesson, we explored the nucleus, the great library and command center of the cell. We learned how it holds the precious DNA blueprint and issues instructions in the form of messenger RNA to govern the cell's life. But instructions, no matter how precise, are only potential. To turn those plans into action, to build, to move, to grow, to live, requires energy. This lesson covers the organelles responsible for generating this energy. We are now moving to the power plants of the cell. We will explore the remarkable organelles known as the mitochondria. You have likely heard mitochondria, that is the plural form. A single one is a mitochondrion, referred to by their famous nickname, the powerhouses of the cell. This is not just a catchy phrase, it is the literal truth. These are the organelles that create the vast majority of the cell's usable energy. Without the work of the mitochondria, the cell would be like a city in a blackout. The instructions from the nucleus would go unread, the factories would fall silent, and the cell's life would cease. Let us first visualize the structure of this vital organelle. A mitochondrion is a small, often oval-shaped structure, but its design is one of beautiful complexity. It is a double-walled organelle, much like the nucleus. It has a smooth outer membrane, which acts as a simple container, separating it from the rest of the cytoplasm. But it is the inner membrane where the true magic happens. This inner membrane is not simple at all. It is extensively folded. These folds are called cristae, and they look like shelves or tunnels extending into the very center of the organelle. These cristae are a brilliant evolutionary design. Their purpose is to dramatically increase the available surface area. If you were to flatten out this inner membrane, it would be many, many times larger than the outer membrane. This vast surface is crucial, as it is studded with the molecular machinery, the tiny enzymes and proteins that perform the final, critical steps of energy production. More surface area means more room for this machinery, and thus a far more efficient power plant. The fluid-filled space inside this folded inner membrane is called the matrix. This matrix is also packed with enzymes, each one responsible for a specific chemical reaction. If you are still awake, picture this structure, a smooth outer boundary, and inside it, a complex, folded inner boundary, creating a massive workspace for the business of energy. So, how does this power plant work? The primary function of the mitochondria is to perform a process called cellular respiration. This is the process of converting fuel into a form of energy the cell can actually use. Think of it like a familiar power plant in our world. A plant might burn coal or natural gas, using oxygen, to generate electricity. The mitochondrion does the same, but on a molecular scale. The fuel it burns is glucose, a simple sugar that comes from the food you eat. The oxygen it uses is the very oxygen that you breathe in with every breath. Inside the cell, the glucose is first broken down into smaller pieces in the cytoplasm. These smaller pieces are then imported into the mitochondrion, into its matrix. Here, in a complex series of chemical reactions, the fuel is broken down even further. It is burned in a controlled, step-by-step process using the oxygen you inhaled. The exhaust from this process is carbon dioxide, which you then exhale, and water. The very act of breathing is a direct connection to the work of these tiny organelles. You breathe in the oxygen they need, and you breathe out the carbon dioxide they produce as waste. But what about the electricity? What is the useful product? The energy released from burning glucose is not just released as heat that would be wasteful and would cook the cell. Instead, the mitochondria capture this energy and store it in a special rechargeable molecule. This molecule is the most important energy-carrying molecule in all of life. It is called ATP, which stands for adenosine triphosphate. You can think of ATP as the cell's universal energy currency. It is the dollar of the cell. Or perhaps, a better analogy, is a tiny, rechargeable battery. When the cell needs to perform a task, contract a muscle, send a nerve signal, or build a new protein, it doesn't use glucose directly. It pays for the task with an ATP molecule. The ATP molecule releases a small, perfectly sized burst of energy by breaking one of its chemical bonds, turning into ADP, adenosine diphosphate, a used battery. The entire purpose of cellular respiration in the mitochondria is to take that used ADP and, using the energy from glucose and oxygen, recharge it. It sticks that third phosphate group back on, turning it back into a fully charged ATP molecule ready to be spent again. It is a constant, rapid cycle of spending and recharging, happening billions of times per second in every one of your cells. Let us pause for a moment to consider a fascinating detail, a deep look into the history of life. Mitochondria are unique. They are the only organelles outside of the nucleus to have their very own DNA. Their DNA is small, circular, and very similar to the DNA of bacteria. This discovery led to the powerful endosymbiotic theory. This theory suggests that mitochondria were once free-living, independent bacteria billions of years ago. At some point in the distant past, a larger host cell engulfed one of these energy-producing bacteria. But instead of digesting it, a symbiotic relationship formed. The host cell provided protection and food, and the bacterium provided a massive surplus of energy in the form of ATP. Over eons, this bacterium became a permanent, essential part of the host cell, evolving into the mitochondrion we know today. They are ancient guests, the descendants of once-free organisms, living within our cells and powering our existence. This brings us to the real-world relevance of these power plants. The energy you use to live, to think, to move, and even to rest is being produced, right now, by the mitochondria in your cells. Cells that do more work have more mitochondria. Your muscle cells are packed with thousands of them, ready to provide the ATP needed for contraction. Your brain cells are also incredibly dense with mitochondria, as thinking is a very energy-intensive process. The steady beat of your heart is powered by mitochondria. When you feel energetic, it is because your mitochondria are efficiently meeting your body's energy demands. When you feel fatigued, it is often because the demand for ATP is outpacing the supply. We have now established a system. We have the instructions, the master plans, secured in the nucleus, and we now have the energy, the power source, generated by the mitochondria in the form of ATP. We have the plans and the power. But what are we building? The cell must now use this energy to follow the nucleus's instructions. It must build its own components. It must manufacture the proteins, the enzymes, and the structures of life. The next logical step in our journey is to visit the cellular factories. With energy produced, the cell needs a system to read the instructions and build the products. This is the role of the ribosomes and the vast network they live on, the endoplasmic reticulum. Our next lesson will focus on these tiny protein factories, the ribosomes. But for now, just rest. Feel the quiet, constant work of your own mitochondria, the ancient power plants within you, steadily and calmly powering your body as you drift. Ribosomes, the protein factories. Welcome back. In our quiet progression through the cell, we have visited the centers of control and power. In our last lesson, we explored the mitochondria, the remarkable power plants that provide a constant, reliable supply of ATP, the cell's energy currency. And before that, we explored the nucleus, the secure command center that holds the DNA blueprint and sends out work orders in the form of messenger RNA or mRNA. We have now arrived at a critical junction in our journey. We have the plans, the mRNA instructions, and we have the power to the ATP energy. But plans and power are static. They do nothing on their own. The cell now needs the workers. It needs the builders. It needs the actual machines that can take those instructions and, using that energy, build the components of life. This lesson is dedicated to these tireless, essential, and universal workers, the ribosomes. These are the protein factories of the cell. Let us first understand what a ribosome is. A ribosome is not an organelle in the same way as the nucleus or the mitochondrion. It is not enclosed by a membrane. Instead, a ribosome is a molecular machine. It is an incredibly complex, intricate piece of machinery, one of the oldest and most fundamental in all of life. Each ribosome is constructed from two main components. It is made of a special type of RNA called ribosomal RNA, or rRNA, and a collection of specific proteins. This ribosomal RNA is not a message like mRNA. Rather, it is a structural and functional component. It forms the very scaffold and core of the machine, acting as the framework upon which the proteins are assembled to create the final, functional unit. Every ribosome consists of two distinct parts, or subunits. There is a large subunit and a small subunit. For most of the time, These two subunits float separately in the cell. Only when a new set of instructions arrives, that strand of messenger RNA from the nucleus, do these two subunits come together, clamping down on the mRNA strand like a vice, ready to begin their work. If you are still awake, picture this tiny machine. It is not alive. It is a pure automaton. Its purpose is singular. Its function is protein synthesis. This is, perhaps, the most important single fact to remember. Ribosomes make proteins. Why is this so important? Because proteins are the stuff of life. They are not just for building muscle. They are the workers of the cell. They are the enzymes that run chemical reactions. They are the structural components, like collagen, that hold you together. They are the hemoglobin that carries oxygen in your blood. They are the channels and pumps in the cell membrane. They are the antibodies that fight infection. The cell is what its proteins do, and every single one of them is built by a ribosome. These tiny factories are found in all living cells, from the simplest bacterium to the most complex human neuron. This universality speaks to their ancient, essential nature. They are a fundamental requirement for life as we know it. Inside our eukaryotic cell, we find ribosomes in two main locations. Some are free ribosomes. They float unattached in the cytoplasm. These free-floating factories build proteins that are destined to be used within the cytoplasm itself. Other ribosomes are bound ribosomes. They are physically attached to the surface of a large, folded membrane network, which we will explore in our next lesson. These bound ribosomes give that network a rough appearance, and they are responsible for building proteins that are destined to be exported from the cell or embedded in the cell's membranes. But how does this factory work? This is the central, beautiful process of life. The ribosome's job is to read the genetic message and translate it into a new physical form. This process is, therefore, called translation. The analogy of an automated assembly robot is most fitting, or perhaps a ticker tape reader from an old telegraph office. The ticker tape is the strand of messenger RNA that arrives from the nucleus. This tape is a long sequence of 4 chemical letters, which we call bases. The message is written in 3-letter words called codons. Each codon, a sequence of 3 bases, is a specific instruction. It is the code for one specific amino acid. Amino acids are the building blocks of proteins. Think of them as a set of 20 different types of Lego bricks. By linking them together in different sequences, you can build a countless variety of structures. So, here is the process. Let it play out in your mind. The 2 subunits of the ribosome come together and clamp onto the mRNA strand. The small subunit reads the codons. The large subunit builds the protein. The ribosome reads the first 3-letter codon, let's say AUG. This codon is a start signal, and it also codes for a specific amino acid. The ribosome now needs that amino acid. But how does the correct brick arrive? The cell uses another helper molecule, a small, specialized delivery truck called transfer RNA, or tRNA. Each tRNA molecule is designed to do one thing. It picks up one specific type of amino acid from the cytoplasm, and it carries it. On its other end, the tRNA has a 3-letter key called an anticodon that matches the mRNA's lock or codon. So, the ribosome reads AUG. The only tRNA molecule whose key matches AUG floats in, docks at the ribosome, and delivers its specific amino acid. The ribosome then, mechanically, shuffles down the mRNA tape by exactly 3 letters to the next codon, let's say CGC. The ribosome now waits. A new tRNA molecule, the one whose key matches CGC, arrives, carrying its specific amino acid. Now, the ribosome performs its most crucial action. Acting like a welder, the ribosome's large subunit takes the new amino acid and links it with a chemical bond called a peptide bond to the first amino acid. A chain has begun. The first, now empty tRNA truck is released, and it goes off to find another amino acid. The ribosome shuffles down again. It reads the next codon. The correct tRNA arrives. The ribosome links the new amino acid to the growing chain. This is the steady, rhythmic work of the ribosome. Read, attach, link, shift. Read, attach, link, shift. Codon by codon, amino acid by amino acid, the ribosome stitches together a long chain, a polypeptide chain, in the exact sequence prescribed by the DNA. This continues on and on until the ribosome reads a special stop codon on the mRNA. This signal tells the ribosome that the protein is finished. The entire complex, the ribosome, the mRNA, and the new protein chain all fall apart. The ribosome subunits are free to find a new message. The protein chain, newly born, is now floating free. This process is the direct link between your genes and your physical reality. Every protein in your body was, at one time, a thought in the mind of the DNA, a message carried by RNA, and a physical product assembled by a ribosome. But the ribosome's job ends here. It has built the chain. However, this chain, this long, floppy string of amino acids, is not yet a functional protein. It is just a string. A protein's power, its function, comes from its unique, complex, three-dimensional shape. It must be folded like a piece of molecular origami into its precise final form. How does this folding happen? And how does this new protein get to where it needs to go? For the proteins built on the bound ribosomes, the story is just beginning. As they were being built, they were being threaded into the very membrane network they were attached to. This leads us to our next lesson. We must now explore this vast cellular workshop, this interconnected network of membranes where new proteins are folded, modified, and packaged for their final destination. Our next lesson will explore the endoplasmic reticulum. For now, rest. Let your mind settle on the quiet, constant hum of trillions of tiny factories within you, reading the code of life and building your very being, one protein at a time. The endoplasmic reticulum, the cellular workshop. We are moving deeper into the cell's intricate living machinery. Our journey has brought us to a place of creation. In our last lesson, we focused on the ribosomes, the tiny, universal machines that translate the genetic code, building the long, raw chains of amino acids that will become proteins. We learned that some of these ribosomes float free, but many others are bound to a vast internal membrane network. Our next stop is this very network. Welcome to the cell's great manufacturing plant, its sprawling internal workshop. This is the endoplasmic reticulum, a name that simply means network within the cytoplasm. We often shorten it to just the ER. This is not a small, simple organelle. The ER is a massive, three-dimensional labyrinth of membranes that fills a large portion of the eukaryotic cell. It is a system of interconnected, flattened sacs and branching tubes called cisternae. The space inside these tubes and sacs, which is separate from the general cytoplasm, is called the lumen. In fact, the membrane of the endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope. You can picture it as an extensive, branching highway system that begins right at the wall of the central office, the nucleus. This vast network is so central to the cell's function that it is divided into two distinct yet connected regions. These two regions are defined by the very presence or absence of the ribosomes we just discussed. First, let us examine the rough endoplasmic reticulum, which we call the RER. It is named rough simply because under an electron microscope, its surface is studded with those bound ribosomes, giving it a beaded or rough appearance. This is where the cell's most sophisticated manufacturing begins. As a ribosome on its surface builds a new protein chain, it does not release it into the cytoplasm. Instead, it threads that chain directly into the lumen, the internal space of the RER. Think of it as a factory worker, the ribosome, building a product and feeding it directly onto a factory's main assembly line, the RER. Once inside the RER lumen, this new protein chain, this long string of amino acids, is no longer just a string, its true work begins. Here, it is met by other helper proteins, called chaperones, which assist it in its most critical task, folding. A protein's function is dictated entirely by its precise, complex, three-dimensional shape. An unfolded protein is as useless as an unbuilt machine. Inside the RER, this folding begins. The protein chain twists, bends, and contorts into its specific, stable, and functional shape. But the work of the RER doesn't stop at folding. It is also a modification plant. Many proteins, especially those destined to interact with the outside world, need upgrades. The RER adds other molecules to them. A common process is glycosylation, where a short sugar chain is attached, turning the protein into a glycoprotein. These sugar tags are like labels or badges that will later help identify the protein or help it fold correctly. The RER is also a quality control center. It inspects the new proteins it has folded. If a protein is misfolded or damaged, the RER gives it a chance to refold. If it fails, the cell has a system to identify this defective product, mark it, and send it for destruction. Only correctly folded, properly modified, and inspected proteins are allowed to move on. The second region of this network is the smooth endoplasmic reticulum, or SER. It is called smooth because it lacks ribosomes, giving its surface a clean, tubular appearance. The SER is a different kind of workshop. If the RER is the heavy assembly line for proteins, the SER is a specialized chemical production plant. Its functions are diverse and vary greatly depending on the cell type. One of its primary jobs is lipid synthesis. This is where the cell manufactures its fats and oils. This includes the phospholipids that are essential for building and repairing all of the cell's membranes, including the plasma membrane itself. The SER is the membrane factory. It also synthesizes steroids. In your body, cells in the adrenal glands, or the testes and ovaries, which are responsible for making steroid hormones, like cortisol or testosterone, are packed with an enormous amount of smooth ER. Another critical function of the SER is detoxification. It contains a collection of powerful enzymes that can modify and neutralize harmful substances. These enzymes can make toxic, fat-soluble drugs or poisons into water-soluble substances that the body can then easily excrete. As you might guess, the liver cells in your body, which are the primary detoxification centers, are filled with vast networks of smooth ER. Finally, the smooth ER acts as a vital storage depot. It specifically sequesters and stores calcium ions. It actively pumps calcium out of the cytoplasm and holds it within its lumen. When the cell needs it, a signal can trigger the SER to open its calcium channels, releasing a flood of calcium back into the cytoplasm. This release of calcium is a critical on switch for many cellular processes. In your muscle cells, for instance, the SER, which is given a special name, the sarcoplasmic reticulum, stores the calcium that, when released, triggers the very act of muscle contraction. So, we have these two sections of one vast network. The RER, a rough-surfaced assembly line, folding and modifying proteins, and the SER, a smooth-surfaced chemical plant, building lipids, detoxifying poisons, and storing calcium. After these proteins and lipids have been manufactured, folded, and modified, they are not yet at their final destination. They are still inside the ER. The cell must now prepare them for shipping. The ER packages these finished products. Small, membrane-bound sacs called transport vesicles bud off from the edges of the ER, carrying the newly made proteins and lipids inside. These vesicles are like small, temporary cargo containers that have been loaded at the factory. But where do these containers go? They are not yet ready to leave the cell or go to their final workstation. They must first go to the central sorting, packaging, and shipping center of the cell. They must be sent to the post office. Once these proteins and lipids are made in the workshop, they must be sorted, tagged with their final address, and packaged for final delivery. This is the job of our next organelle. Our journey now takes us from the factory floor to the shipping department. Our next lesson will explore the Golgi apparatus. For now, just rest. Let your mind absorb this concept of the ER. A vast, internal highway system, a protein folding factory, and a chemical synthesis plant, all working in the quiet, dark, and busy center of the cell. The Golgi apparatus, the cellular post office. Welcome back. As you continue to rest, as you allow your mind to drift in this quiet space of learning, we will follow the path of the molecules we have just seen created. In our last lessons, we visited the cell's great workshop, the endoplasmic reticulum. We saw the ribosomes on the rough ER building the raw chains of proteins, and we saw them threaded inside to be folded and modified. We learned of the smooth ER building lipids and handling detoxification. And we saw how both of these factories packaged their finished goods, their new proteins and lipids, into small, membrane-bound sacs. These are the transport vesicles. They are the cargo containers, freshly loaded at the factory. But where do these containers go? They are not yet ready for their final job. They are like unfinished products, or bulk goods, that must be sorted, refined, and labeled for their ultimate destination. These vesicles now travel from the ER to our next stop, the cell's central distribution hub. In this lesson, we examine this remarkable organelle, the cell's post office, its sorting and shipping center. This is the Golgi apparatus, also known as the Golgi complex or the Golgi body. Let us first build a picture of its structure. The Golgi apparatus is, like the ER, an organelle made of membranes. However, it is not a single, continuous, net-like structure. Instead, the Golgi is a neat, orderly stack of flattened, membrane-bound sacs. These individual, flattened sacs are called cisternae. If you are still awake, you can picture the Golgi as a stack of pancakes, or perhaps more accurately, a stack of pita breads. Each cisterna is a separate, hollow compartment, not physically connected to the others in the stack. A typical cell may have only a few of these stacks, while a cell that is specialized in shipping things out may have hundreds. The Golgi apparatus is a highly dynamic and polarized structure. This means it has a clear indoor and a clear outdoor. The side of the Golgi stack that faces the endoplasmic reticulum is the receiving dock. This is called the cis face. The transport vesicles that budded off from the ER travel through the cytoplasm and arrive here. They fuse with the membrane of the first cisterna and empty their contents, their cargo of proteins and lipids, into the lumen, the internal space of the Golgi. Once inside, these molecules begin a journey through the Golgi stack. They are passed from one cisterna to the next, as if moving down a processing line. As they travel, the Golgi's primary work is performed. This is the central concept. The Golgi apparatus's function is to modify, sort, and package these molecules. It is the finishing and sorting center. The modifications that began in the ER are now completed and refined. New, complex sugar chains might be added or existing ones trimmed. Other molecules might be attached. Proteins are finalized, inspected, and made ready. As the molecules reach the shipping side of the Golgi, they arrive at the trans face. This is the outdoor of the post office, the side that faces away from the nucleus and toward the rest of the cell. Here, the Golgi performs its final and most critical task: sorting. How does the Golgi know where each of these thousands of different proteins and lipids is supposed to go? It reads their molecular tags. These tags are specific chemical sequences, like the sugar chains we mentioned, that act like a postal code or a shipping label on a package. The Golgi is, in essence, a sophisticated sorting machine. It recognizes these tags and gathers all the molecules heading to the same destination. It then bundles this shipment together. The membrane at the transface begins to bud off, creating a new, fresh vesicle. This vesicle, loaded with its specific, sorted, and finalized cargo, is now ready for delivery. Let us explore this process a little deeper. The analogy of the post office, first used by the biologist George Palladay, is almost perfect. Raw mail, vesicles from the ER, arrives at the receiving dock, the sys face. The mail is processed and a zip code is added, or read, chemical modifications. The mail is then sorted into different bags for different delivery routes, sorting at the transface. Finally, the mail bags, the new vesicles, are loaded onto delivery trucks to go to their final addresses. This intricate, stacked structure was first described in 1897 by the Italian physician Camillo Golgi. He was studying the nervous system and, using a new staining technique that involved silver, he saw what he called an internal reticular apparatus within the nerve cells. It was a net-like structure, dark against the light background, clustered around the nucleus. For decades, many scientists believed this Golgi complex was simply an artifact, a trick of the staining chemical. But in the 1950 seconds, with the invention of the electron microscope, its existence was finally, definitively confirmed. We now know it is a central, vital organelle in all eukaryotic cells. The real-world relevance of this organelle is profound. It is the very heart of the cell's secretory pathway. This is the pathway that allows cells to make and release substances into the outside world. Think about the cells in your pancreas that are responsible for producing the hormone insulin. Insulin is a protein. It is built on the ribosomes of the RER. It is folded and then sent to the Golgi. In the Golgi, it is processed and packaged into vesicles. When your body signals that your blood sugar is high, these vesicles are instructed to move to the cell's outer membrane, fuse with it, and release their cargo of insulin into your bloodstream. This process is called secretion. The same is true for the cells in your salivary glands, which secrete digestive enzymes, or the cells in your airways, which secrete mucus. Any cell that exports a large number of products, especially proteins, will be filled with a large and active Golgi apparatus. This demonstrates how the cell is a cohesive, flowing system. The nucleus holds the plans, the ribosome builds the product, the ER folds and modifies it, and the Golgi packages and ships it. It is a seamless, coordinated assembly line, turning a genetic instruction, a thought in the DNA, into a functional product that can affect your entire body. But as we've seen, the Golgi is the central shipping center. It ships products outside the cell, as in the case of insulin. It also ships new proteins to the cell membrane itself, to repair it or to install new channels and receptors. But the Golgi also creates specialized packages that are meant to stay inside the cell, to be used for internal affairs. What if the package itself is the final product? What if the cargo is not a helpful hormone, but a set of powerful, dangerous, digestive enzymes? What if the cell needs a stomach of its own to break down waste or unwanted invaders? The Golgi packages these, too, into a special, reinforced vesicle. This particular package is not for export. It is the cell's own internal recycling and waste disposal center. This leads us to our next lesson. We will now investigate these specialized packages, the cell's cleanup crew, known as the lysosomes. For now, just rest. Let the concept of this silent, internal post office settle in your mind. a place of sorting, of finalizing, and of shipping. The Golgi apparatus. Lysosomes and peroxisomes, waste disposal. We are continuing our journey, following the intricate pathways of the cell. In our last lesson, we visited the Golgi apparatus, the cell's master post office. We saw how it receives, modifies, sorts, and packages proteins and lipids into vesicles, sending them to their final destinations. We learned that many of these packages are destined for export, to be secreted from the cell. Others are sent to repair the cell's own membrane. But now, we will follow a different kind of package. We will look at a special vesicle, created by the Golgi, that is not designed for export. It is a package meant for internal use only, and it is filled with a very powerful cargo. This lesson covers the cell's cleanup crew, its waste disposal and recycling system. A living cell, like any busy city, produces waste. It has old, worn-out parts that need to be removed. It may take in food that needs to be broken down, and it must defend itself against foreign invaders. To handle these tasks, the cell employs a specialized organelle. Welcome to the lysosome. The word lysosome itself means digestive body. Lysis means to break apart, and soma means body. This is a perfect name for it. A lysosome is a simple, spherical, single-membrane sac. It is essentially a small bag that has budded off from the Golgi apparatus. But what makes it special is what it contains inside. The lysosome is filled with an array of powerful hydrolytic enzymes. These are proteins that are capable of breaking down, or digesting, almost any large biological molecule. They can dismantle proteins, nucleic acids, like DNA and RNA, carbohydrates, and lipids. They are the cell's ultimate demolition crew. This, of course, presents a danger. If these powerful, destructive enzymes were to leak out, they would begin to digest the cell itself. The cell would be destroyed from within. So, how does the cell protect itself? It does so with a brilliant two-part security system. First, the enzymes are safely contained within the lysosome's own membrane, a sturdy wall that keeps them separate from the rest of the cell. Second, and even more clever, these enzymes are designed to only function in a highly acidic environment. The lysosome's membrane contains active protein pumps that are constantly pumping hydrogen ions into the lysosome, making its internal pH about 5. This is roughly the acidity of a tomato. The rest of the cell, the cytoplasm, is neutral, with a pH of about 7.2. If you are still awake, picture this. The lysosome is a hot zone for digestion. Its enzymes are on and active in the acid. If the lysosome were to break and its enzymes were to spill out, they would find themselves in the neutral cytoplasm, and they would immediately deactivate. They simply cannot work in that environment. This is the cell's safety mechanism. So, what does the lysosome do with this contained power? It has 3 primary jobs. The first is digestion. In single-celled organisms like an amoeba, this is how they eat. The amoeba engulfs a piece of food, creating a food vacuole. This vacuole, a sac containing the food, then fuses with a lysosome. The enzymes pour onto the food, breaking it down into small, useful molecules, sugars, amino acids, that the cell can absorb and use as fuel. Your own immune cells, specifically white blood cells, do this exact same thing. When a white blood cell encounters a bacterium, it eats it, a process called phagocytosis. It then fuses that bacterium with its lysosomes to destroy it completely. The second job is perhaps the most important for the cell's daily life. This is autophagy, which literally means self-eating. A cell's components, its organelles, do not last forever. A mitochondrion might become old, damaged, or inefficient. The cell must recycle it. In this process of autophagy, the cell forms a membrane around the old, worn-out organelle, quarantining it. This sac then fuses with a lysosome. The enzymes flood in and dismantle the entire mitochondrion, breaking it down into its basic raw materials. These materials, amino acids, lipids, fatty acids, are then transported back out into the cytoplasm, where they can be used to build a new, healthy mitochondrion. The lysosome is not just a waste dump, it is the cell's ultimate recycling center. It ensures that nothing is wasted. The third job is more drastic: programmed cell death, or apoptosis. Sometimes, a cell is infected beyond repair, or it is no longer needed in the body's development. In these cases, the cell can be given a signal to self-destruct. The lysosomes can be instructed to rupture, releasing all their enzymes at once to scuttle the cell. Now, there is another cleanup organelle, often confused with the lysosome, but very different. This is the peroxisome. Peroxisomes are also small, single-membraned sacs, but they are not formed by the Golgi. They are believed to be self-replicating, growing, and then dividing, much like mitochondria. And they contain a different set of enzymes for a different set of jobs. If the lysosome is the heavy-duty demolition and recycling plant, the peroxisome is the specialized chemical treatment plant. Peroxisomes have two main functions. The first is to break down very long chains of fatty acids. These long chains are difficult for the cell to use. Peroxisomes chop them up into smaller 2-carbon pieces, which are then sent to the mitochondria to be used as fuel. Their second, and more famous, function is detoxification. Peroxisomes are found in large numbers in your liver and kidney cells. They are specialized in neutralizing harmful substances, most notably alcohol. But their most defining feature is how they handle oxidative stress. Many of the chemical reactions inside the peroxisome produce a highly reactive and dangerous byproduct, hydrogen peroxide, H2O, $2. This is a toxic molecule that can cause immense damage to the cell. The peroxisome's design is a marvel of efficiency. It not only produces this toxic H2O, $2, but it also contains the enzyme to deal with it. It is packed with an enzyme called catalase. Catalase's one and only job is to instantly grab that toxic hydrogen peroxide and convert it into two harmless substances: water, H, $2O, and oxygen, O, $2. The peroxisome handles a dangerous chemical reaction by containing both the reaction and its own antidote in one tiny compartment. The real-world importance of these organelles is immense. When the lysosome fails, the results are tragic. In Tay-Sachs disease, for example, a single, specific lysosomal enzyme is missing. This enzyme is needed to break down a particular lipid in the brain. Without it, this lipid builds up and builds up, accumulating inside the nerve cells until the cells become clogged with this waste and die. So, we have the lysosome for digestion and recycling, and the peroxisome for fatty acids and detoxification. We have now covered the major metabolic organelles, the parts of the cell that manage energy, production, and waste. But the cell is not just a bag of these chemical factories. It needs shape, it needs structure, it needs a way to move. How does the cell maintain its form? How does it transport vesicles from the ER to the Golgi? How does it crawl or move? To understand this, we must examine the cell's internal framework. Our next lesson will explore the bones and muscles of the cell, the cytoskeleton. But for now, just rest. Let your mind be at ease, knowing that these silent, efficient, and powerful systems are always at work, keeping your cells clean, balanced, and healthy. The cytoskeleton, internal support and movement. Welcome back. As we continue our exploration of the cell's inner world, we have now visited a great number of its specialized organelles. We've seen the nucleus, the command center, the mitochondria, the power plants, the ribosomes and the factories, and the Golgi, lysosomes and peroxisomes, which act as the cell's shipping and sanitation departments. It is easy, with this knowledge, to picture the cell as a simple, bag-like container, a watery sac called the cytoplasm, where all of these organelles are just floating. But this picture is incomplete. A cell is not just a formless blob, it has a definite shape. It can be a long, branching nerve cell, a flat, tile-like skin cell, or a biconcave disc like a red blood cell. How does a cell maintain its specific shape? How does it prevent itself from collapsing into a simple puddle? And how does it move? When a vesicle buds off the ER, how does it know how to get to the Golgi? It does not just drift by chance. This brings us to our next crucial lesson. In this module, we will explore the cell's internal framework, its bones, and its muscles. This is the cytoskeleton. The cytoskeleton is a dynamic, intricate, and ever-changing network of protein fibers that extends throughout the entire cytoplasm. It is far more than just a static scaffold. It is a system that provides structural support, but it also acts as the cell's muscles for movement and its highway system for internal transport. This network is one of the most defining features of the eukaryotic cell. To understand the cytoskeleton, we must break it down into its three main components. These are three distinct types of protein filaments, each with its own structure and its own set of vital jobs. Let us begin with the thinnest of these fibers, the microfilaments. These are solid rods composed primarily of a protein called actin. Actin is an incredibly common and important protein in eukaryotic cells. The microfilaments are often found in a branching network just inside the plasma membrane. Here, their job is to bear tension. Think of them as a system of internal guy wires or flexible ropes that help the cell resist pulling forces and maintain its overall shape. They give the cell's cortex, or outer layer of cytoplasm, a semi-solid, gel-like consistency. But the microfilament's most famous role is in movement. Actin filaments are the muscles of the cell. They can create movement by interacting with another protein, a motor protein called myosin. The most dramatic example of this is in your own muscle cells. Your muscle tissue is packed with an enormous, highly organized arrangement of actin and myosin filaments. When your muscle contracts, it is not because the fibers themselves shorten. It is because the myosin motors crawl along the actin filaments, causing them to slide past one another. This collective sliding, happening billions of times at once, is the physical basis for every movement you make. This same principle allows cells themselves to crawl. In a process called amoeboid movement, the cell can rapidly assemble actin filaments at its leading edge, pushing the membrane forward in a structure called a pseudopod, or false foot. It's a dynamic, flowing motion, as if the cell is pulling itself along. Your own white blood cells use this very method to crawl out of your blood vessels and chase down infections. Now, let us consider the next component, the steel cables of the cell. These are the intermediate filaments. They are intermediate in size, thicker than microfilaments but thinner than the next component we will discuss. Unlike the other two types, intermediate filaments are not defined by a single protein. They are a diverse family. The protein keratin, for example, which makes up your hair, nails, and the tough outer layer of your skin, is an intermediate filament. The defining feature of intermediate filaments is their durability. They are the cell's most permanent and stable structural elements. Their job is pure, mechanical reinforcement. If microfilaments are like ropes for tension, intermediate filaments are like the steel rebar in concrete. They are tough, and they are built to last. Their main function is to anchor things in place and resist mechanical stress. They form a sturdy cage around the nucleus, holding it in its proper position. They extend throughout the cytoplasm, keeping other organelles from drifting. In skin cells, they are essential for giving the tissue its strength, preventing it from tearing when stretched. They are the true, permanent scaffolding of the cell. Finally, we come to the largest and, in many ways, the most complex of the cytoskeletal elements. These are the microtubules. Microtubules are hollow tubes, like tiny rigid pipes, constructed from a protein called tubulin. Microtubules have two major roles. First, like girders in a building, they are responsible for resisting compression. They help the cell maintain its shape, especially against pushing forces. But their most dynamic and fascinating role is as the cell's internal transportation system. The microtubules form a vast network of railway tracks that crisscross the cell. And along these tracks, tiny motor proteins act like locomotives. These motor proteins, with names like kinesin and dynein, bind to the microtubule track with one foot, and they hold a package, like a transport vesicle from the ER with their other arm. If you are still awake, picture this in your mind. A motor protein, energized by ATP, literally walks, step by step along a microtubule, dragging its cargo. This is how the vesicle from the ER finds the Golgi. It is how neurotransmitters are moved from the center of a nerve cell all the way down its long axon. It is a highly organized, directed, and essential logistics network. Microtubules also have a critical role in large-scale movement. They are the structural core of two organelles of motion, cilia and flagella. Cilia are short, hair-like projections that beat in unison, like tiny oars, to move fluid past a cell. The cells lining your windpipe, for example, use cilia to sweep mucus and debris up and out of your lungs. A flagellum is a much longer, whip-like tail, and it's used to move the entire cell. The most famous example is the flagellum of a sperm cell, which propels it on its journey. And, in one of the most beautiful processes in all of biology, microtubules form the mitotic spindle during cell division. When a cell needs to divide, it assembles a complex scaffold of microtubules. These fibers attach to the condensed chromosomes and, like a set of microscopic pulleys, they carefully pull the identical copies of DNA apart, ensuring that each new daughter cell receives a perfect, complete set. So, the cytoskeleton is not just a single thing. It is a dynamic, multi-part system. It is the microfilament muscles, the intermediate filament, scaffolding, and the microtubule highways. We have now explored almost every component inside the cell. We have examined the organelles and the very framework that holds them. We have a complete picture of the city and its infrastructure. There is only one major component left to discuss. We must now zoom out and examine the city wall itself. The skin of the cell that encloses all of this, that separates the living world inside from the non-living world outside. Our next lesson will explore the flexible, functional, and all-important barrier, the plasma membrane. But for now, rest. Let your mind be still, supported by this new knowledge, just as the cell is supported by its own intricate internal framework. The plasma membrane, the gatekeeper. Welcome back. As you rest, as your mind remains open, we continue our journey. We have now traveled deep into the inner world of the cell. We have seen the nucleus with its plans, the mitochondria creating energy, the factories of the ER and ribosomes, the shipping center of the Golgi, the recycling plants of the lysosomes, and even the very bones and muscles of the cytoskeleton that give the cell its shape and its structure. We have explored, in detail, the entire living city. But we have not yet discussed the city wall. We have not explored the boundary, the barrier that separates the complex, organized, living world inside the cell from the chaotic, non-living environment outside. In this lesson, we will study one of the most vital and elegant structures in all of biology. We will examine the plasma membrane. This is the skin of the cell. Every single living cell, from the simplest bacterium to the most complex human neuron, is defined by this boundary. It is not a rigid, static wall. It is a structure that is flexible, dynamic, and incredibly intelligent. It is the gatekeeper. It must contain all the cell's precious components, but it cannot be a sealed box. Life requires a constant exchange with the outside world. Nutrients must come in, waste must go out, and signals must be received. The plasma membrane performs all of these tasks with amazing precision. To understand how it works, we must look at its structure. Our modern understanding of this structure is called the fluid mosaic model. This name is a perfect two-word description of what it is. Let's break that down. The fundamental structure, the fabric of the membrane, is the phospholipid bilayer. This is the fluid part. A phospholipid is a special kind of lipid, or fat molecule. It has a very specific, two-part personality. It has a head, which is hydrophilic, meaning water-loving. This head is electrically charged and is attracted to water. And it has two long tails, which are hydrophobic, meaning water-fearing. These tails are fatty, have no charge, and are repelled by water. Now, picture what happens when you drop millions of these molecules into water, which is the environment both inside and outside the cell. They spontaneously organize themselves. They form a double layer, a bilayer. The water-loving heads all turn to face the water, forming a surface on the outside of the cell and another surface on the inside of the cell. The water-fearing tails have nowhere to go but away from the water. So, they all turn inward, huddling together in the middle, creating a dry, oily, water-free core. If you are still awake, visualize this. A sandwich where the bread is the water-loving heads and the filling is the water-fearing tails. This bilayer is the essence of the membrane. It is what makes it a barrier. Because its center is oily and hydrophobic, it prevents water-soluble things, like salts and sugars, from simply passing straight through. It is a soap bubble, a flexible, self-sealing container. But this fluid barrier is not the whole story. This is where the mosaic part comes in. The membrane is a mosaic because it is studded with a diverse collection of other, larger molecules, primarily proteins. These proteins are like the functional pieces, the gates, sensors, doors, and anchors that are embedded within the fluid lipid sea. Some of these proteins are integral proteins. They pass all the way through the entire membrane, with one end sticking out of the cell and the other end sticking in. These proteins are the true gatekeepers. They form channels that can open and close, creating a specific tunnel for a single substance, like a sodium ion, to pass through. Or they are pumps, which use energy to actively move substances from one side to the other. Other proteins are peripheral proteins. They are not embedded in the membrane, but are just loosely attached to one side or the other, often acting as switches or anchors for the cytoskeleton. And there are other molecules in this mosaic. Cholesterol is dotted throughout the membrane, where it acts as a fluidity buffer. It keeps the membrane from becoming too fluid at high temperatures or too rigid and brittle at low temperatures. And finally, on the outer surface of the cell, you will find short, branching chains of carbohydrates. When these chains are attached to proteins, we call them glycoproteins. When they are attached to lipids, we call them glycolipids. Let's dive deeper into this fluid mosaic concept. When we say fluid, we mean that the membrane is not a solid structure. The individual phospholipid molecules, and many of the proteins, are free to drift sideways, to move around. They are like people milling about in a crowded room. This fluidity is essential for the membrane's function. This brings us to the real-world relevance of this remarkable structure. This skin is not just a passive container, it is the cell's face to the world. It is the primary site of communication. How does one cell talk to another? How does your brain tell your muscles to move? How does a hormone, released far away, have an effect on a target cell? The answer is on the membrane. Those integral proteins, the sensors, are called receptor proteins. A hormone, like insulin, circulates through your blood. It is a message, but it never enters the target cell. Instead, the insulin molecule docks onto a receptor protein on the outside of the cell membrane, one that has the perfect matching shape. This docking is like a key fitting into a lock. When the key turns, it changes the shape of the protein inside the cell. This shape change triggers a cascade of chemical reactions within the cell, a domino effect. That tells the cell, the message has arrived, please open your gates and let glucose in. The message is passed, but the messenger never crossed the border. And what about those carbohydrate chains? The glycoproteins and glycolipids. They form a sugar coating on the outside of the cell, called the glycocalyx. This coating is like a molecular ID badge or a fingerprint. It tells the rest of the body who this cell is. Your cell's sugar coating is unique to you. This is the very basis of your immune system. When your white blood cells are on patrol, how do they know which cells to attack and which to leave alone? They read these ID badges. They check the glycoproteins. If they read a badge that is self, they move on. But if they find a cell with a foreign badge, like a bacterium, or a virus-infected cell, or a cell from a transplanted organ, they recognize it as non-self and launch an attack. The plasma membrane is the boundary between self and non-self. It is the gatekeeper of all traffic and the face of all communication. We have now established that the plasma membrane is a selective barrier. It is selectively permeable. This is its defining function. It controls, with absolute precision, what enters the cell and what leaves. But how? We have mentioned channels and pumps. But how do things get across this barrier, especially if they don't have a special gate? How does water move? How does the cell pump things against their will? We have defined what the barrier is. Our next logical step is to understand how it is crossed. This is the subject of our next lesson, cellular transport. But for now, just rest. Let your mind float in this idea of a fluid, living boundary, a soap bubble with a purpose, a gatekeeper that defines the very edge of life. Cellular transport, moving materials. Welcome back. As you continue to rest, we return to the very edge of the living cell. In our last lesson, we explored the plasma membrane, that remarkable, fluid mosaic that defines the cell's boundary. We learned that it is a gatekeeper, a flexible, intelligent, and selectively permeable barrier. It is selective because it does not and cannot let just anything pass through. We have now arrived at the central question of this boundary. How? How does the cell import the nutrients it needs to live? How does it export the waste it produces? How do things actually get across this barrier? This lesson explains the diverse and elegant mechanisms of cellular transport. We will explore the rules of the road, the ways materials move from one side of the membrane to the other. This entire concept is governed by one of the most fundamental forces in nature, a force that requires no energy at all. This is the tendency for things to move from an area of high concentration to an area of low concentration. Imagine you have a drop of ink. If you place it in a glass of still water, the ink does not stay in one spot. It spreads out, it diffuses. It moves from the area where it is highly concentrated, the drop, to the areas where it is not, until it is evenly distributed. This spreading out is driven by the random motion of molecules, and it is a universal law. This difference in concentration, from high to low, is called a concentration gradient. You can visualize it as a hill. It is natural and requires no effort for a ball to roll down the hill from a high point to a low point. It is the same for molecules. This downhill movement is the basis for the first and simplest category of transport, passive transport. Passive means it requires no energy from the cell. The cell just lets it happen. The simplest form of this is called simple diffusion. Some very small, non-charged molecules can pass directly through the oily, lipid core of the membrane. The most important examples are the gases oxygen, $O, $2, and carbon dioxide, $CO, $2. If you are still awake, picture this. Your blood carries a high concentration of oxygen to your cells. The inside of your cell, which has used its oxygen, has a low concentration. The oxygen, following its concentration gradient, simply and silently diffuses downhill from your blood through the membrane and into your cell, no energy required. At the same time, your cell, as a waste product, has a high concentration of carbon dioxide. The blood has a low concentration. So, the $CO, $2 diffuses out of the cell, downhill and into the blood to be carried away. Simple, passive, and perfectly efficient. But what about water? Water is also a small molecule, and it is essential for life. The diffusion of water, specifically, across a selectively permeable membrane, has its own special name, osmosis. This is a critical concept. Water, just like the ink, will also move from an area where it is highly concentrated pure water to an area where it is less concentrated, water that has salt or sugar dissolved in it. This is why osmosis is so important to your cells. If you place a red blood cell in pure, distilled water, the water outside the cell is at a higher concentration than the slightly salty water inside the cell. Water will rush into the cell, downhill, trying to balance the concentrations. It will rush in until the cell swells up and bursts. Conversely, if you place that same red blood cell in extremely salty water, the situation is reversed. The water inside the cell is now at a higher concentration than the very salty water outside. Water will rush out of the cell, downhill, causing the cell to shrivel and die. This is precisely why drinking salt water is so dangerous. It literally pulls the water out of your cells. What about all the other molecules? What about glucose, the cell's main fuel? Or ions, like sodium and potassium, which are charged and cannot pass through the oily membrane? For these molecules, the cell provides a gate. This is still a form of passive transport, because the molecules are still moving downhill, from high to low concentration. But because they need help, we call it facilitated diffusion. Facilitated just means helped. This help comes from those integral proteins we discussed, the ones embedded in the membrane. There are two types. Channel proteins are simple tunnels. They open up, and a specific ion, like a potassium ion, can flow through, following its gradient. They are fast and efficient. Carrier proteins are more complex. They are like a revolving door. A glucose molecule, for example, will fit into a special pocket on the carrier protein. This docking causes the protein to change its shape, flipping the glucose molecule to the other side of the membrane, where it is released. Again, no energy is used. It is just a downhill roll, but through a specific, protected gate. So, simple diffusion, osmosis, and facilitated diffusion, they all are forms of passive transport. They are all rolling downhill. But what if the cell needs to push something up the hill? What if a cell needs to hoard a resource to pull in a molecule that is already more concentrated on the inside? What if it needs to actively push out a waste product, even if the concentration outside is high? This is where the cell must spend its energy. This is active transport. Active transport is the process of moving molecules against their concentration gradient. It is pushing the ball up the hill, and this, of course, requires energy. The energy it uses is that universal battery, ATP, the molecule made by the mitochondria. Active transport is performed by specific proteins in the membrane called protein pumps. These pumps are true machines. They use the energy from an ATP molecule to grab a specific substance, change their shape, and force that substance to the other side, against its will, uphill, from low concentration to high concentration. The most famous example is the sodium-potassium pump. This pump is vital for your entire nervous system. It actively pumps sodium out of your nerve cells and potassium into your nerve cells, both against their gradients. This builds up a steep hill, an electrical and chemical charge. This charged state is like a ready signal, and when the nerve fires, it is this very hill of ions that comes rushing down, creating the electrical impulse of your thoughts. Finally, what if the cell needs to move something big? What if it needs to eat a whole bacterium or secrete a large bundle of hormones? The substance is far too large for any pump or channel. This is where the cell uses bulk transport. This involves the membrane itself. To bring something in, the cell uses endocytosis, or in-cell action. The membrane itself sinks inward, surrounding the particle, until it pinches off, forming a new vesicle inside the cell. If the cell is eating a solid particle, like a bacterium, we call it phagocytosis, cell eating. If it is drinking a pocket of the surrounding fluid, we call it pinocytosis, cell drinking. To move something out, it does the reverse. This is exocytosis, or out-of-cell action. This is the process we saw with the Golgi. A vesicle, filled with a product like insulin, travels to the plasma membrane. The vesicle's membrane fuses with the plasma membrane, like two soap bubbles joining. This fusion opens the vesicle to the outside, and its contents are expelled, or secreted, from the cell. These are the mechanisms, from the silent, passive diffusion of oxygen to the complex, energy-hungry work of the protein pumps. The cell is a master of managing its own borders. We have established that the membrane is a selective barrier, and we have seen how animal cells cross it. But so far, we have focused primarily on the structures common to animal cells. A vast kingdom of life. The kingdom of plants builds its cells with a major, fundamental difference. A difference that completely changes how the cell deals with one of these transport methods, especially osmosis. Plants have a rigid, strong outer layer that animal cells lack. Our next lesson will explore the unique structures of plant cells, beginning with this defining feature, the cell wall. But for now, just rest. Let these concepts of movement and balance settle in, the constant, quiet, and regulated flow of life across the cellular border. Plant cell structures, the cell wall. Welcome back. As you continue to rest, we begin a new and important chapter in our exploration of the cell. Thus far, our journey has focused, for the most part, on the structures and functions of the animal cell. We explored its dynamic, flexible boundary, the plasma membrane. We learned in our last lesson how that membrane controls the flow of materials and how it must carefully manage the process of osmosis, lest the cell burst from taking in too much water or shrivel from losing it. The animal cell, in all its complexity, is just one expression of eukaryotic life. We now turn our attention to another, vast and ancient kingdom, the kingdom of plants. Plant cells, at their core, share much with animal cells. They have a nucleus, they have mitochondria and ribosomes and an endoplasmic reticulum. They have a Golgi apparatus and a plasma membrane. But to survive, a plant must solve a different set of problems than an animal. A plant cannot move to find food. It cannot run from danger. It cannot seek shelter from the environment. And it must, above all, hold itself up against the constant pull of gravity, often to great heights. To meet these unique challenges, the plant cell has evolved a set of remarkable, specialized structures. And our lesson begins with the most defining, most obvious, and most rigid of them all. We begin not at the plasma membrane, but outside of it. We begin with the cell wall. Let us be perfectly clear. the plant cell has a plasma membrane, just like the animal cell. It is still the gatekeeper, the living, selective boundary that controls what truly enters the cell's cytoplasm. But in a plant cell, this delicate membrane is not the outer boundary. It is, instead, encased, unprotected, and supported by a thick, strong, and rigid outer layer. This is the cell wall. If the animal cell's membrane is a flexible soap bubble, the plant cell's membrane is a soap bubble built inside a strong, precisely shaped cardboard box. The primary function of this wall is structural support and protection. It is this wall that gives the plant cell its fixed, often rectangular or box-like shape. And it is this wall that, when multiplied by trillions, allows a plant to grow from a soft, green shoot into a towering, strong tree. What is this wall made of? Its primary component is a remarkable molecule, one of the most abundant and important organic molecules on our planet. It is called cellulose. Cellulose is a polysaccharide, a long, complex chain made of thousands of glucose, or sugar, molecules, all linked together. These long cellulose chains are bundled together like steel cables, forming incredibly strong fibers called microfibrils. These fibers are then woven, layer upon layer, into a mesh that forms the cell wall. When a plant cell is young and still growing, it secretes a thin, flexible primary wall. This wall is strong, but it is also elastic enough to allow the cell to expand and elongate. Once the cell has reached its mature size, it often builds a much thicker, more rigid secondary wall on the inside of the primary wall. This secondary wall is heavily reinforced and is what gives wood its incredible, lasting strength. Now, let us return to the problem of osmosis, which we discussed in our last lesson. If you are still awake, recall what happens to an animal cell, like a red blood cell, if it is placed in pure, distilled water. The water, following its concentration gradient, rushes into the cell, and the cell, with its flexible membrane, swells and swells until it bursts, or lyses. Now, let's perform that same experiment with a plant cell. We place it in pure water. Just as before, water rushes into the cell, again, following the laws of osmosis. The cell's internal water bag, which we will discuss later, begins to swell. The cell's plasma membrane begins to push outward, but it does not burst. It cannot. As the membrane pushes out, it presses against the unyielding, rigid cell wall. The wall is strong enough to resist the pressure. It pushes back against the cell with an equal and opposite force. This internal, water-driven pressure against the cell wall is one of the most important concepts in plant biology. It is called turgor pressure. This turgor pressure is, in essence, what keeps a plant inflated. When a plant is well watered, its cells are full. They are turgid. They are pressing firmly against their walls. This collective, internal pressure is what allows a soft-stemmed plant, like a flower, to stand upright. It is the hydrostatic skeleton of the plant. And what happens when you forget to water that plant? You have seen the result. The plant wilts. This is not because the cell walls have failed. It is because the turgor pressure has been lost. The cells have lost water to the air. The internal water bag has shrunk. The membrane has pulled away from the cell wall, and the cells are no longer inflated. The plant goes limp. The moment you add water, osmosis resumes, the cells refill, turgor pressure is restored, and the plant stands up again. The cell wall, therefore, is the plant's brilliant solution to living in a watery world. It allows the cell to actively draw in water, to be full of water, without the danger of bursting. This turgor pressure is the very life force of the plant's structure. This brings us to the real-world relevance of this one structure. The cellulose in plant cell walls is all around us. The fiber in your diet, the parts of vegetables, fruits, and grains that your body cannot digest is, quite simply, the cellulose from the plant cell walls. Your body lacks the enzyme to break down those cellulose chains. Wood, the material we have used to build our homes and fires for millennia, is little more than the accumulated, dead, secondary cell walls of tree trunks. The strength of a giant redwood is the collective strength of these cellulose fibers. The cotton in your clothes is nearly pure cellulose. The paper this knowledge might once have been written on is processed, refined cellulose. We have now seen how the cell wall provides the plant with its structure, its shape, and its solution to the problem of osmosis. This rigid box is a perfect adaptation for a stationary life. But this solution creates a new, profound problem. If you are locked in a box, if you are anchored in the ground, how do you get food? An animal can move to find its food. A plant cannot. It must, therefore, make its own. This leads us naturally to our next lesson. We will explore another organelle unique to plants, the specialized solar power plant that does what no animal cell can. We will explore the chloroplast, the site of photosynthesis. But for now, just rest. Let the concept of this strong, rigid wall settle in, a wall of cellulose, bracing the cell and holding the plant up to the sky. Plant cell structures, chloroplasts, and photosynthesis. Welcome back. As you rest, as your mind remains open, we continue our exploration of the plant cell. In our last lesson, we explored the cell wall, the rigid, structural barrier that gives the plant cell its shape and its strength. We learned that this wall is a brilliant adaptation, allowing the cell to absorb water and build up turgor pressure without the danger of bursting. But this solution, this life lived inside a rigid box, defines the plant's stationary existence. An animal can move, it can hunt or forage or find a new source of fuel. A plant is anchored in one place. This simple, profound fact leads to a fundamental question. If a plant cannot move to find its food, how does it get the energy to live? How does it acquire the fuel for its own mitochondria? This lesson covers the plant cell's remarkable and life-giving solution. We will now explore the organelle that powers, quite literally, nearly all life on Earth. We are going inside the chloroplast. If the mitochondrion is the cell's power plant, converting fuel into energy, the chloroplast is the solar power plant. It is the only organelle that can create that fuel in the first place, using the most abundant energy source on our planet, sunlight. The function of the chloroplast is singular and monumental. It is the site of photosynthesis. Photosynthesis is the chemical process of capturing light energy, the energy from the sun, and converting it into stable, storable chemical energy in the form of sugar. It is the process of making food from light. The overall chemical recipe for this process is one of elegant simplicity in its summary, though infinitely complex in its execution. The chloroplast takes three simple non-living ingredients: carbon dioxide, $CO, $2, which it draws from the air; water $H, $2O, which is pulled up from the roots, and the energy from sunlight. Using these three ingredients, the chloroplast performs a series of chemical reactions to create two products. The first is glucose $C, 6H, 12O, $6, a sugar, which is the plant's food, its stored chemical energy. And the second, which is a waste product for the plant, is oxygen $O, $2. Let us now look at the structure of this solar factory. A chloroplast is a large lens-shaped organelle, and just like the nucleus and the mitochondrion, it is a double-walled organelle, enclosed by both an outer membrane and an inner membrane. Inside this double boundary is a dense, fluid-filled space called the stroma. This is like the cytoplasm of the chloroplast. It is within this stroma that the synthesis, part of photosynthesis, will happen, the building of the sugar. But floating within this stroma, we find the real machinery, a third, light-independent membrane system. This is a system of interconnected, flattened, membrane-bound sacs. A single sac, or disc, is called a thylakoid. You can picture it as a hollow pancake. These thylakoids are often stacked neatly on top of one another, like a pile of coins. A single stack of thylakoids is called a granum, or grana for plural. And embedded within the membrane of these thylakoids, like tiny solar panels, is the most crucial molecule, the pigment chlorophyll. Chlorophyll is the molecule that gives plants and leaves their characteristic green color. It is green because it is absorbing all the red and blue wavelengths of light for energy, and it is reflecting the green light, which is why we see it. This pigment is the very antenna that physically absorbs the light energy from the sun. The process of photosynthesis occurs in two main stages, each in a different part of the chloroplast. The first stage, the light-dependent reactions, happens in those thylakoid membranes. Here, the chlorophyll captures solar energy and, using water, converts that energy into small, temporary, high-energy battery molecules, like ATP. This is also where water is split and oxygen is released as a byproduct. The second stage, the light-independent reactions, also called the Calvin cycle, happens in the stroma, the fluid outside. Here, the chloroplast uses the energy from those battery molecules, and, in a complex chemical cycle, it takes carbon dioxide from the air and uses it to build the stable, long-term energy storage molecule, glucose. And here, as we look deeper, we find another fascinating echo of the past. Just as we did with the mitochondrion, chloroplasts also contain their own separate circular DNA, and they contain their own ribosomes. This is overwhelming evidence for the endosymbiotic theory. Just like the mitochondrion, the chloroplast is believed to be the descendant of a once free-living, independent bacterium. Specifically, a cyanobacterium, which was a bacterium that had already invented photosynthesis. Billions of years ago, a larger host cell engulfed this bacterium. Instead of being digested, it formed a symbiotic partnership, trading its food-making ability for a safe, stable home. It is another ancient guest, living within the cell and powering our entire world. Now, as we are discussing the unique features of the plant cell, we must mention one other dominant structure that works in concert with the cell wall. While the animal cell has small temporary vacuoles, the mature plant cell has a central vacuole. This is a single, enormous, membrane-bound sac that can take up to 90% of the entire cell's volume. It is, in essence, a giant water bag. This central vacuole is the cell's main storage tank. It holds a watery solution called cell sap, and it stores not just water, but also nutrients, pigments, and even defensive toxins or waste products. But its most important role is to maintain turgor pressure. By filling with water through osmosis, this giant vacuole swells. It presses the cytoplasm and the plasma membrane outward, pushing firmly against the rigid cell wall. It is the central vacuole, working with the cell wall, that keeps the plant cell turgid, firm, and upright. The real-world relevance of the chloroplast and photosynthesis is, quite simply, everything. It is not an exaggeration to say that this single process is the foundation of our entire biosphere. First, it is the food. Photosynthesis is the base of virtually every food chain on the planet. The energy you get from eating an apple is the stored solar energy, captured by chloroplasts and turned into sugar. The energy you get from eating meat comes from the animal that ate the grass, and the grass was powered by chloroplasts. All life, with just a few small exceptions, runs on this captured sunlight. Second, there is the matter of the air you are breathing as you rest. The oxygen in our atmosphere, the gas that is essential for our own cellular respiration in our own mitochondria, is a byproduct of photosynthesis. For billions of years, these tiny, green, solar-powered organelles have been silently capturing sunlight and releasing the oxygen that has shaped our planet and made our own complex life possible. We have now explored the fundamental structures of the animal cell, and we have just examined the unique, defining features of the plant cell: the rigid cell wall, the massive central vacuole, and the brilliant solar engine of the chloroplast. We now have all the pieces. In our next lesson, we will formally synthesize this knowledge. We will place these different blueprints side by side. We will create a clear comparison, reviewing what they share and what makes each of them a unique solution to the challenge of being alive. We will also revisit our very first topic, the simple prokaryote, to complete our picture of life's foundational designs. But for now, just rest. Let these concepts settle. The plant cell, a strong, self-sufficient, and quiet creator, making food and air from light and water. Comparing cell types, a synthesis. Welcome back. Our journey through the intricate world of the cell has been a long and detailed one. We have traveled, lesson by lesson, through the inner workings of life's fundamental unit. We have explored the nucleus as the command center, the mitochondria as the power plants, and the chloroplasts as the solar engines. We have seen the factories, the shipping centers, the recycling plants, and the very framework that holds the cell together. We have studied the blueprint of the animal cell. We have studied the unique blueprint of the plant cell. Now, in this review module, our task is to synthesize this knowledge. It is time to step back from the individual components and to look at the blueprints themselves, side by side. This is a moment to organize the information that has settled in your mind, to see the elegant patterns of life, and to understand why these different, beautiful forms exist. Let us begin with the simplest and most ancient design, the one we mentioned in our very first lesson, the prokaryotic cell. The name itself, from the Greek pro, meaning before, and karyon, meaning kernel or nucleus, tells us its defining feature. These are the before nucleus cells. Organisms like bacteria are prokaryotes. Their structure is one of profound, successful simplicity. They possess no nucleus. Their genetic material, a single, circular strand of DNA, floats freely within the cytoplasm, in a region we call the nucleoid. More than that, the prokaryotic cell lacks all the complex, membrane-bound organelles we have spent so long discussing. There are no mitochondria, there is no endoplasmic reticulum, there is no Golgi apparatus. A prokaryotic cell is a single, open room, a small, efficient workshop where all the tools and all the machinery are in one place. It is a masterpiece of efficiency, but it is, by design, simple. Now, let us turn to the other great design, the eukaryotic cell. From the Greek eu, meaning true, these are the cells with a true nucleus. This is the blueprint for all complex multicellular life that you see. All plants, all animals, all fungi. The eukaryotic cell is a city of compartments. Its defining feature, the one that sets it apart, is the nucleus. This membrane-bound vault protects the cell's precious DNA, allowing for a much larger and more complex set of instructions, but the complexity does not end there. The eukaryotic cell has a vast division of labor, which we have been exploring. It has specialized, membrane-bound organelles. It has the mitochondria to handle energy. It has the ER and ribosomes for complex manufacturing. It has the Golgi for shipping. This compartmentalization, this separation of tasks into different rooms, allows the eukaryotic cell to achieve a level of complexity and specialization that is simply impossible for the prokaryote. It can become much larger, and it can cooperate with other cells to build vast, intricate organisms, like a tree, or a bird, or a human. But even within this eukaryotic blueprint, we have, in our lessons, focused on two major variations, two different solutions to the problem of being a complex, living thing. First, there is the eukaryotic animal cell. This is the cell we used as our primary model. It is defined by its flexibility. It has the nucleus, the mitochondria, the the Golgi, the lysosomes. But just as important is what it lacks. It has no cell wall. Its boundary is the flexible, dynamic plasma membrane. This flexibility allows the animal cell to move, to change its shape, to crawl using its cytoskeleton, and even to engulf other things, a process we called phagocytosis. It also has no chloroplasts. The animal cell is heterotrophic, other feeding. It must find and consume its fuel from the outside world. And second, we have the eukaryotic plant cell. It shares that same fundamental eukaryotic chassis. It has the nucleus, the mitochondria, the the Golgi, but it has been adapted for a completely different life. It is stationary, and it is autotrophic, or self-feeding. To solve the problems of this lifestyle, it has three unique structures that we have just discussed. It has the cell wall, a rigid box of cellulose, built outside the plasma membrane to provide the structural support that an animal gets from a skeleton. It has the chloroplasts, the green solar engines that allow the plant to ignore the problem of finding food and to instead make its own, using only sunlight, water, and air. And it has the large central vacuole, the enormous water-filled sac that presses against the cell wall, creating the turgor pressure that keeps the plant firm and upright. So, we have these three great designs. If you are still awake, you can use a simple mental checklist to distinguish them. When you look at any cell, the first question is always, does it have a nucleus? If the answer is no, you are finished. It is a prokaryote. If the answer is yes, you know it is a eukaryote. You then ask the next set of questions. Does it have a rigid cell wall? Does it have chloroplasts? Does it have a large central vacuole? If the answer is yes to these, you are looking at a plant cell. If the answer is no, if its boundary is a flexible membrane, you are looking at an animal cell. It is just as important, in this synthesis, to remember what all cells have in common. Despite these vast differences in complexity and strategy, all three of these blueprints, the bacterium, the plant cell, and the animal cell, share a core set of components. This shared hardware is a powerful testament to a single, shared origin of life, deep in the Earth's distant past. First, all cells have a plasma membrane. Every single living cell must have this selective skin to separate its internal, living chemistry from the non-living world outside. Second, all cells have cytoplasm, the jelly-like fluid that fills the cell and in which all the components are suspended. Third, all cells use DNA as their genetic material, the blueprint for life. In the eukaryote, it is safely stored in the nucleus. In the prokaryote, it floats in the nucleoid. But the language, the molecule, is the same. And fourth, all cells have ribosomes. This is a profound and telling fact. The small, non-membrane-bound machines that build proteins are universal. The nucleus, the mitochondria, the chloroplasts, these are options in life's design. But the plasma membrane, the DNA, and the ribosomes, these are requirements. Why is this distinction, this simple separation of prokaryote versus eukaryote, so critically important? It is not just an academic detail. It is, quite literally, a matter of life and death. This is the entire foundation of modern antibiotic medicine. When you have a bacterial infection, a prokaryotic infection, a doctor prescribes an antibiotic, like penicillin. These drugs are selective poisons. They are engineered to attack structures or processes that are unique to prokaryotic cells. Penicillin, for example, works by attacking and breaking the linkages in the bacterial cell wall, a structure that your eukaryotic cells do not have. Other antibiotics, like streptomycin, are designed to gum up the specific prokaryotic version of the ribosome, shutting down their protein factories. The medicine is therefore toxic, even lethal, to the invading bacteria, but it leaves your eukaryotic cells, with your different ribosomes and complete lack of a cell wall, completely unharmed. This selective toxicity is one of the greatest achievements of medicine, and it is based entirely on understanding this fundamental evolutionary divide. We have now completed our grand comparison. We have laid the blueprints side by side. We have defined the prokaryote, the animal cell, and the plant cell. We have seen what makes them different, and we have seen what makes them all life. We have, in essence, examined every part in great detail. But a cell, any cell, is not just a static list of parts. It is not just a blueprint. It is a living, breathing system. The parts do not exist in isolation. They are in constant communication, in a dynamic, flowing dance of energy and information. For our final lesson, let us step back one last time. Let us put all these components into motion and reflect on the cell not as a collection of things, but as a single, living, and harmonious system. Conclusion: the cell as a system. We have reached the end of our journey. This is our final lesson. Over the course of our study, we have taken the living cell, this fundamental unit of life, and we have disassembled it piece by piece. We have done this in a quiet, methodical way, just as a watchmaker might lay out the components of a fine timepiece. We have visited the great library of the nucleus and the tireless, ancient power plants of the mitochondria. We have stood on the factory floor of the endoplasmic reticulum and watched the ribosomes building the very machinery of life. We have followed the packages to the Golgi apparatus, the central post office, and we have observed the cleanup crews of the lysosomes and peroxisomes. We have felt the bones and muscles of the cytoskeleton, and we have stood at the city wall, the plasma membrane, and watched the complex dance of cellular transport. We have compared the blueprints, the prokaryote, the animal cell, and the plant cell, with its unique wall and chloroplasts. It is tempting, now, to see the cell as this list of parts, a collection of organelles, a bag of stuff. But this final lesson, perhaps the most important of all, is to put all of these pieces back together. It is to see that the cell is not a list of things, but a relationship between them. It is to see the cell not as a collection of parts, but as a single, dynamic, living system. This is the central concept. The cell is the fundamental unit of life, because it is the system that is alive. No single part of the cell is alive on its own. Your DNA stored in the nucleus is just a chemical molecule. It is not alive. A mitochondrion, if removed from the cell, is just a small bag of enzymes. It is not alive. A ribosome is a machine, no more alive than a tiny, complex robot. Life, the quality of being alive, is an emergent property. This is a concept where the system as a whole, the whole cell, has properties and abilities that the individual components do not possess on their own. The system, the organized, interacting, and collective whole, is far, far greater than the sum of its parts. If you are still awake, picture this. Imagine a car. Is the engine a car? No. Is a wheel a car? No. Is a seat a car? No. But when you assemble all of these non-car parts in a very specific, coordinated way, a new property emerges, the property of transportation. The cell is the same, the property of life, the ability to metabolize, to respond, to grow, to reproduce. This property emerges only when all these non-living parts work in perfect harmonious concert. Let us trace the flow of one single action to see this system in motion. Imagine your body needs to release a hormone, a protein messenger, like insulin. The journey does not begin at the part that makes it. It begins with a need. First, a signal, perhaps another hormone, arrives from the outside world. It docks with a specific receptor protein on the plasma membrane, the gatekeeper. This key in the lock changes the protein's shape inside the cell. This one action triggers a cascade, a chain of information that travels to the nucleus, the command center. The nucleus finds the specific gene for insulin, the recipe in the vast DNA library. It then transcribes this recipe into a photocopy, a strand of messenger RNA. This whole process, this high-level management, requires immense energy. So, the mitochondria, sensing the demand, are constantly at work, burning glucose, using oxygen, and churning out a steady supply of ATP batteries, which are sent to power the nucleus and every other part of this story. The mRNA message now leaves the nucleus, passing through a nuclear pore. It finds a bound ribosome on the surface of the rough ER. The ribosome locks on and begins translating the message, building the raw chain of the insulin protein and feeding it directly into the ER's internal space. Inside the ER, the protein chain is met by chaperones. It is folded into its correct, complex 3D shape. It is modified. Once it passes quality control, it is packaged into a transport vesicle. This vesicle now needs to travel. It is picked up by a motor protein, a tiny machine that, powered by more ATP, walks along the railway tracks of the cytoskeleton. It walks from the ER to the receiving dock of the Golgi apparatus. The vesicle fuses. The protein enters the Golgi, where it is finalized. It is further modified, sorted, and tagged with its final address. It is now a finished product. The Golgi, at its shipping dock, packages this finished insulin into a new secretory vesicle. This new vesicle, again, travels along the cytoskeleton, a guided journey, to the inner side of the plasma membrane. There, it waits for the final go signal. When the signal is given, the vesicle fuses with the plasma membrane, a process of exocytosis. It opens to the outside world, and the finished, functional insulin hormones pour into your bloodstream, ready to do their job. In this one, single story, in just a few seconds, we have seen the nucleus, the ribosome, the ER, the Golgi, the cytoskeleton, the plasma membrane, and the mitochondria, all working in a seamless, coordinated, and beautiful symphony. While this was happening, lysosomes were busy recycling old, worn-out proteins to provide the raw amino acids for this new one. The membrane's pumps were busy maintaining the correct ion balance, and osmosis was keeping the cell's water pressure stable. This is the cell, not a list of parts. It is a flowing, living river of information and energy. It is a state of perfect, dynamic balance, a state we call homeostasis. The true relevance of all this is you. As you lie here, resting, you are not a single thing. You are a community. You are a vast, cooperative society of an estimated 30 trillion of these living, breathing systems. Every single aspect of your existence, every thought that flickers through your mind, every memory you hold, every beat of your heart, every simple, unconscious breath you take, is the summed, collective, and harmonious expression of these trillions of cellular systems, all working within you. You are this symphony. You have now completed your foundational lesson on the cell and its organelles. This knowledge, these stories of the inner world, are now settling into your mind. They are becoming a part of your own understanding, a new way to see the living world and to see yourself. Our educational journey will continue, but for now, your work is done. You can let these concepts rest, as you, yourself, now rest. As you drift deeper, your mind can organize and review this new knowledge. For now, sleep well.